this is where we left it last week when we discussed somewhat briefly that when eukaryotic cells splice introns out of their pre-mRNA molecules or primary transcripts, sometimes they do it in such way that all of the introns are taken out, but most of the times they will show some sort of alternative splicing. The names for these various forms of alternative splicing have to do with what happens to introns or exons. So for instance, exon skipping is when a whole exon is skipped and taken out from the primary transcript. Mutually exclusive exons is a version of that in which different combinations of exons make it to the final mature mRNA and so forth. And we discussed briefly that the way cells do this is by seeing or not certain combinations of splice donor and acceptor sites. Whenever the spliceosome needs to take a portion, notice I'm not saying intron anymore, a portion of a pre-mRNA out of the molecule, they use a donor site, a branching point, and an acceptor site, and they see those things through base complementarity to special small non-coding RNAs that they carry within them. So the spliceosome, this machine that does splicing, carries in it these small nuclear or small nucleolar RNAs that will allow them to see by base complementarity these donor or acceptor sites. Okay, so I want you to picture that in your heads. There is this machine that carries these small non-coding RNAs in them that they use to read these donor and acceptor sequences in pre-mRNA molecules. And then whatever combination of donor and acceptors they see in one of those pre-mRNA molecules is what will allow them to take out a chunk of sequence from those pre-mRNAs. Whether that chunk is just an intron or whether it may, like during exon skipping, include an exon, that kind of depends on what that cell is able, or that spliceosome is able to quote unquote see in that pre-mRNA molecule. Okay? Or read. Did that make sense? So alternative splicing comes down, alternative splicing is not so much an inherent property of the pre-mRNA molecule. It's not the pre-mRNA molecule that says which way it should be spliced. Is a property of the cell that is expressed in certain spliceosomes that have the ability to see and read all of the donor and acceptor sites, or may skip some of them. Okay? All right, so the question now is, and this is the question that I'm asking of all of you, how can a cell not see a donor or acceptor site? Or let's rephrase it, how can a spliceosome miss one of these in a pre-mRNA molecule? Blocked by a protein. Love that. And Emily. I'm gonna get there, I promise. So Emily says maybe some of these donor or acceptor sequences in the pre-mRNA are blocked by a protein. Proteins that bind RNA? Yeah? Okay. So blockage, I can't spell today. Blockage by RNA binding protein. Cool. Any other ideas?
Weak signals?
Weak signals, what do you mean by that?
They're
They're
not communicating.
not communicating. Okay, so you're saying that there is a weak signal. I'll take it, but then you rephrased it and you said they're not communicating. How do these donor and acceptor sites and the spliceosome communicate with one another? What mechanism do they use? You had the instinct there. I'm gonna give you. What do you think? So how are spliceosomes and pre-mRNAs communicating with each other? You went blank, Ale?
Like base pair.
The base pairing that we just mentioned, right? So the way the spliceosomes read these things in pre-mRNA molecules is through base pairing. So can base pairing be weak? Can base pairing ever be weaker or stronger? I see some heads shaking no, some heads shaking yes.
Maybe it's like biased.
Bias how?
Like if the first one is not as favorable, it'll skip it in favor of the other.
Oh, okay, so in a way we're revisiting this idea that maybe that first donor site is not as favorable, as in it cannot create that strong base pairing, and it will energetically or dynamically or probabilistically will skip that one and go for the second, right? Is that more or less what you had in mind? Kind of, when you were saying that, yeah, base pairing can be made weaker, what did you have in mind?
Well, you can have certain sequences that have more CG regions, which have more hydrogen bonds to the carbon.
Okay, and
Mutation.
mutations. So let me write some Jacob and
Urea.
Medea. Elaborate on that, the mutations idea. So mutations where?
Like in the base pairing?
In the base pairing, so but the mutation would be on the pre-mRNA or on the spliceosome side?
Oh.
Or both? On the pre-mRNA, right? I kind of saw with the back of my eye a hand up somewhere here, or am I seeing visions?
And then relating to the strength, would like something like the Kozak sequence need like a specific set of sequences, and determining from those consensus, you determine the strength of each one of those sequences?
Okay, so we're fine-tuning it a little further. Yes, so the idea here is not so much about CG content, which will increase the strength, but it's more about complementarity, right? And that's, in a way, the point, that you can have weaker or stronger reading of the donor and acceptor sites depending on the extent of complementarity between those donor and acceptor sequences in the pre-mRNA and those small non-coding RNAs that the spliceosome carries with it, right? It's almost like the spliceosome will carry with it a lens that is tailored for a specific sequence, but it might still see a sequence that it's mismatching by a pair or two or a letter or two, right? So that can still create enough of base pairing that the spliceosome might see it, but it can also cause a weakening of that interaction such that it may skip it at times. And I think, like you, Jacob, said, right? That may skip to another one. Cool. So let's put it all down as this poor communication. We'll call it mispairing. And any other ideas of how a spliceosome may not properly see a donor acceptor site? We have proteins blocking those sequences. We have small nuclear or nucleolar RNAs in the spliceosome that are not base pairing perfectly.
Secondary structures.
Ooh, secondary structures. Andre, correct? Yeah? Secondary structures. And I know you have your hand up.
I'm gonna say the shape of the mRNA, like 3D.
Same idea, 3D, right? So, Arcia. Same idea.
Would it be like an SNC element, maybe like blocking the topology of the mRNA?
So SNC as in like as a small non-coding, blocking the topology. As an idea, very interesting. I don't know if any described. So the idea here is maybe instead of a protein blocking these sites, it could be another small non-coding RNA that is blocking these sequences by base pairing. Very interesting, conceptually impeccable idea. I don't know of any known examples of that. But the way that mRNA sometimes hide certain sequences is through by engagement of secondary structures. So I have a slide that relates to that in a way. When we think, if I asked any of you to describe the life of an mRNA in eukaryotic cells, there's a decent chance that you come out with a map that looks something like this. And some of my ex-360ers might recognize the slide. Just a little bit of recycling there. But the way that we think of mRNAs in eukaryotic cells is that there is transcription from DNA, then there is pre-mRNA capping, there will be some pre-mRNA splicing and polyadenylation, and eventually a mature mRNA will be exported into the cytoplasm. Its stability may be controlled in a post-transcriptional manner in the cytoplasm, and at some point there's going to be translation initiation generating a protein. So that's more or less the life of an mRNA that we can think of in eukaryotic cells. This is how we normally think of eukaryotic mRNAs, but there is something fundamentally wrong with this image, right? And what is fundamentally wrong with this image is that this is what mRNAs more likely look like inside of cells. There's two major differences here. Number one, mRNA molecules are rarely, quote unquote, naked in the cell, both inside of the nucleus and in the cytoplasm. They're actually fully decorated by these RNA binding proteins. There's several families of RNA binding proteins. These things exist. And just like there is a whole bunch of proteins that interact with DNA in a sequence-specific manner so that they won't bind DNA just anywhere, there's a whole bunch of RNA binding proteins that interact with RNAs in a sequence-specific manner, right? And sometimes those interactions are such that certain features, as we would say in jargon, so certain sequences of those mRNA molecules are hidden from view now. If you have a massive RNA binding protein that is binding to an RNA at a given location, it may mask an adjacent or neighbor donor or acceptor site. And now the spliceosome can't see them anymore, right? The other thing that is different in this cartoon from the previous one is that these RNAs are full of these hairpin structures, and those are the structures that RNA molecules will adopt in space through secondary structure formation. So they will fold on themselves and engage in intramolecular base pairing. Right? Any of these hairpins will have the RNA going in one direction, making a U-turn, and then coming back in the opposite direction, such that now two stretches of that RNA molecule that may have complementary sequence are facing each other in anti-parallel orientation. And what will RNAs do in that case? Form a mini double helix. Right? It would be a mini, small double helix of RNA, not DNA. And those hairpin structures can very often hide sequences from view from anywhere else outside of that RNA molecule. So if any of these hairpin structures happen to include part or the whole of a donor or acceptor site for splicing, those sequences are now going to be inaccessible. They're going to be invisible to the spliceosome. Okay? So in a way, this cartoon emphasizes two of the ways in which certain donor and acceptor sites may all of a sudden become invisible to spliceosomes that we discussed, the RNA binding proteins and secondary structures. On the other side of the equation, there is what small nuclear RNAs, or small nuclear RNAs, I'm going to combine both in snRNAs, what snRNAs a given spliceosome carries. And the spliceosome is a machine that has a whole bunch of protein components that are always more or less the same. What changes in them is the snRNA that they carry. And those snRNAs are expressed from DNA in a regulated manner. What that means is cells, given their identity and given their current state, can express certain snRNAs and maybe not others. And what that does is it allows spliceosomes to load onto themselves certain snRNAs, but not others. So what I'm saying there is that cells have a regulated manner to determine which snRNAs will be present in spliceosomes, and therefore which donor or acceptor sites that cell can see or not. So this is how mechanistically certain cells decide that they will splice certain pre-mRNAs one way and not the other. One of the textbook examples of alternate splicing is you have a certain gene that is expressed across multiple cell types, and then it turns out that that pre-mRNA is spliced in a given cell type one way, and in a different cell type a different way. So we go back to how would those cells know what to do? Well, it's a combination of some of these mechanisms. It turns out that cell type A might express an snRNA that allows spliceosomes to see a given donor site, but cell type B doesn't express that snRNA. So effectively, cell type B won't be able to see certain donor and acceptor sites and will splice those pre-mRNA molecules whichever other way can. And vice versa. Maybe cell type B expresses snRNAs that cell type A doesn't. Or it can come down to these RBPs. Cell type A may express a bunch of RBPs that don't exist in cell type B or vice versa, and those RBPs would be blocking certain donor or acceptor sites. What we need to bear in mind, though, is that these things, they may come across as a little random and stochastic and, oh, well, whatever cell type A has and whatever cell type B doesn't and vice versa. No, these things are regulated. Cells, through their evolutionary, developmental history, decide, quote unquote, what complements of snRNAs and RBPs they will be expressing, and that in a way dictates their ability to splice pre-mRNAs a certain way and not others. Yeah?
RBPs and the SRNs.
Exactly. So absolutely. So it may come across as like if a cell cannot see a certain donor site, oh, poor cell, it's not seeing that. No, it may be an active, regulated decision that the cell made, if you will, through its developmental history. So a muscle cell may never want to see a certain donor site. That means that it will splice those pre-mRNAs in a way that are completely useless to it. So it's a good thing that that cell doesn't see that donor site. So how can it never see that donor site? Well, for instance, by not expressing the snRNA that would allow the spliceosome to read it.
So the constituted splicing that we've learned
Mm-hmm.
since way back when,
Yeah, yeah.
a very elementary version of the actual splicing that goes.
Correct, and it's one of those things that we do to ease people and learners into the concept. So now we know that most genes are alternatively spliced. Do we know the mechanism, the detailed mechanisms of how it happens in each cell type under each condition? No, of course we don't. But imagine that you're first learning about splicing and they come at you with alternate splicing. So it's like, what? So we ease into the idea by first explaining splicing and throwing around concepts like introns are spliced out of mRNAs. Not entirely untrue, it's just a little incomplete for the sake of simplicity. Because it is true that introns will be taken out, it's just that the truth of the matter is that some cells will take out introns along with some exons maybe, and other cells will take out different introns along with different exons out of the same pre-mRNAs and so forth. But yeah, it's not a bad thing. It's not necessarily a disability of cells. It's a chosen ability, if you will. And of course, I throw around the term chosen very loosely. There's not like, I don't think of cells as sentient beings making intellectual choices, right? But it's just that through their developmental history, they've restricted their potentials to splice certain pre-mRNAs one way and not others. And I can say you took Bio 462 and Philosophy 380. Questions, anyone, or comments? No? All right, so let's move along. Cool, so now we have, in that previous cartoon, I said that the eukaryotic mRNAs, before they get exported into the cytoplasm, they're capped, polyanylated, and spliced, whichever way. And now we have a mature mRNA that makes it into the cytoplasm. The export into the cytoplasm is a less regulated mechanism. Not that it cannot be regulated, but it's a little less prevalent as a regulatory mechanism. And now we have a mature or processed mRNA in the cytoplasm. And that opens yet a new gate for cells to regulate the expression of the corresponding gene by regulating the stability of those RNAs. And all that means is there's going to be competing forces between destroying this RNA or not. And the way that cells can regulate that process is through the use of targets for degradation. I drew them there somewhere in the 3' UTR. You can tell that because it's downstream of the stop codon. So it's in 3' UTR territory. That's where they usually are. They don't have to be always there. There may be targets for degradation somewhere in coding sequence. It's just that it's a little easier for organisms, for species, to keep them in the 3' UTR, because then it doesn't really matter what happens to sequence around them. You can always play around with sequence and move targets for degradation in and out. Whereas if you start messing around with sequence with encoding sequence, you always have to be careful not to break the code, right? And that imposes an evolutionary constraint. So that's why they tend to be in 3' UTR. And we discussed, I know that it seems like an eternity ago now, but we discussed that when there is alternative polyadenylation of mRNAs, one of the major ways in which alternative polyadenylation affects the expression of genes, given that the coding sequence is normally spared through polyadenylation, the way that the life of that mRNA is affected is because longer 3' UTRs open up the possibility for more of these targets for degradation. It's almost like they're putting more targets on their back, if you will, right? I swear, I just thought of that, but it's kind of funny because it's like if you think of the 3' UTR as the backside of the mRNA, it's like they're putting more targets on their back. Huh? All good? Cool. So, how does this work? It turns out that there is these two major families of small non-coding RNAs that we refer to as microRNAs or siRNAs. On the business side of things, on the business end, they're very similar to each other. So what happens is these miRNAs and siRNAs are very small non-coding RNA molecules. They're short. They're somewhere around 20 nucleotides in length. And they get loaded. Now you start seeing this pattern here, right? So just like snRNAs were loaded into spliceosomes, these things get loaded into something called the RISC. And the RISC is an RNA-induced silencing complex, and it's all in the name. It is a complex that will silence the expression of target mRNAs induced by those mRNA or siRNA sequences they have in them. Okay? So if we zoom in and take a closer look at how this happens, in both cases you have these purple spheres or ovals with a short RNA sequence loaded onto them. And siRNAs tend to show perfect base pairing to their target sequences in the 3' UTR of their target mRNAs. Right? And when the RISC complex senses perfect pairing between the small RNA they carry and the target RNA, that triggers a series of enzymatic capabilities in that RISC complex that will make the RISC cleave that RNA. So there is cleavage of the RNA and obviously degradation. So the RISC will cut the RNA and therefore destroy it if there is perfect base pairing between the small RNA that it carries and the target gene. With microRNAs, it's slightly different. MicroRNAs have evolved in such way that they rarely have perfect base pairing to their target sequences in the 3' UTR of mRNAs. They normally show imperfect base pairing. So out of the 20, 21, 22 nucleotides in that miRNA, there may be 1, 2, or 3 nucleotides that are not properly base pairing to the target sequence. The RISC complex can sense that, and it will not trigger that catalytic nucleolytic activity that it has. So it won't cleave the RNA, but it won't leave either. It will just stay there. And because it stays there, it kind of messes things up for the ribosome and the process of translation. Okay? So these mRNA, the RISC is big, so when the RISC is bound to an mRNA molecule, even though it may not be cleaving it because the miRNA is not showing perfect base pairing, it won't let go of it either, and it prevents translation. So there is inhibition of translation. So in a way, ultimately, the outcome, if you will, is more or less the same. That mRNA is not being translated, either because it cannot be translated or because it's being destroyed. Either way, there's no protein coming out of that mRNA. And it's a way that cells have to say, yeah, I know that I produced this mRNA. I know that I put a lot of time and effort into expressing and processing and transporting this mRNA to the cytoplasm, but now I don't want to translate it anymore. Okay? Cool. So let's see, just make sure that I'm going to address all the text here. So miRNAs and siRNAs are both short double-stranded RNA molecules. They start as double-stranded RNA molecules. One of their strands combines with proteins and forms the RISC to start looking for mRNAs with complementary targets. I think we covered all of that. They differ on how they base pair with targets. We cover that as well. So now the question is, okay, they both start as double-stranded RNAs. How come the miRNAs are their own class, siRNAs are a separate class? Where's all of these business of perfect base pairing or perfect matching versus imperfect matching? Where does that come from? And it comes from how these double-stranded RNAs are originally produced. And when it comes to microRNAs, those short double-stranded RNA molecules are produced by transcription from DNA. So miRNAs are coded in DNA. And they have a very peculiar sequence. So they have a sequence such that there is a stretch of nucleotides. This is abbreviated for simplicity, right? But there is a stretch of nucleotides in DNA that is followed by some spacer sequence, and then the same sequence but reverse complementary. What I mean by that is, in this cartoonish example, we have A, G, T, C, C. The reverse complementary would be to look for the complementary bases going in the reverse direction. So GG, GG, A, C, T, A, C, T. Did you guys see it? So it's a stretch of nucleotides followed by some spacer sequence by the same set of nucleotides in reverse complementary orientation. And what happens is when these things get transcribed from DNA, what will the primary microRNA do? Do one of these hairpins, right? It will fold on itself such that the stretch of reverse complementary sequence now is facing each other in anti-parallel orientation. So if this thing, if you guys remember your 5' and 3', what that means is that there is a 5', 3' going on here, and a 3', 5' going on in the opposite branch of this hairpin. And because these sequences were reverse complementary, now in our cartoonish example, the GG will be paired with the CC and the AGT with the ACT. Okay? So these are the primary microRNA molecules. There are enzymes inside of these things happen in the cytoplasm. There are enzymes that can detect these short hairpin structures. The enzyme is collectively known as dicer because it dices up these things. And what dicer will do is cleave off the loop in the hairpin structure and leave off that short double-stranded RNA stretch, one of which strands will be loaded onto RISC. Okay? Now, all of that is mechanistic, and it's all fine. The message, the important take-home message from a regulatory point of view, and we'll actually talk about microRNAs very soon, because I already found some papers that deal with how microRNAs control genes important for Alzheimer's, so I think that we'll be discussing microRNAs soon, is that these microRNAs are produced by the cell through transcription. So these microRNAs are not foreign entities trying to destroy the cell's mRNAs. These are genes that the cell itself is expressing to actively inhibit the translation of some of its own mRNAs. So it's a feedback regulatory mechanism, and if you will, a negative feedback regulatory mechanism. It's the cell saying, yeah, I know that I transcribed a whole bunch of protein coding genes, made them into mRNAs. Now I will actively express this miRNA gene that will go inhibit some of those or inhibit their translation. So it's the cell saying, at some point I wanted to express this protein, but I don't want to express much of it anymore. Yeah? With siRNAs, the story is a little different. siRNAs originate by cleavage of much longer double-stranded RNA molecules that may be present in a cell. So just like dicer recognizes these short double-stranded stretches present in those miRNA or primary mRNA hairpins, dicer can also see or detect longer double-stranded RNA molecules present in the cytoplasm and chop them up. We know that DNA is a very long double-stranded RNA, double-stranded helix, right? And we also know that RNA can, at times, adopt these very localized, specific secondary structures that we call hairpins. That is true as well. But where will you find long double-stranded RNA molecules in nature? What are those? Do you know of any natural entity that will have very long double-stranded RNA molecules? Maybe virus. When in doubt, just say virus. It's a good chance that you'll be right. Yeah, correct. So an entire family of viruses that are double-stranded RNA molecules, right? So when eukaryotic cells find very long double-stranded RNA viruses in their cytoplasm, immediately they evolve to, quote-unquote, think virus. So what do? They send dicer to chop them up, and then they evolve these very protective mechanism by which one of those short strands in each of these short double-stranded segments of RNA gets loaded onto RISC and used to patrol the cytoplasm for any sequence or any mRNAs that might show complementarity to them. And it's so elegant, so poetic. I don't know if you guys see it right away, but the idea here is if these short RNA sequences that RISC uses to patrol the cytoplasm came from an RNA virus, what RNA molecules will the RISC complex find first? The virus itself, right? So it's almost like the cell generates a whole bunch of, phony IDs, if you will, then loads risk complexes with them, and then it uses those to patrol the cytoplasm. And the chances are that the first thing they'll find is those original double-stranded RNA molecules that now they can destroy. Because one thing that is missing from this cartoon here, and it matters, is that this double-stranded RNA virus is producing a whole bunch of mRNAs that co-opt the translational machinery of the cell that they infected to make viral proteins that will allow them to make more viral molecules that will allow them to infect other cells. Right? So that's all a virus wants to infect a cell and make many copies of itself. Some viruses will do it through genetic information coded in DNA that they will first transcribe to RNA and then translate into protein to make viral proteins. But some viruses, like these double-stranded RNA viruses, they don't need to worry. They use RNA-based polymerases that will make RNA out of RNA, and they will make many mRNAs, if you will, no classic transcription involved, and they will just make viral proteins that way. But because the cell chopped up some of those viral proteins, particles, and made a whole bunch of short siRNAs that can immediately see them, it's a protective mechanism that cells evolved such that whenever they're infected by these type of RNA viruses, they can fight back quickly. And the reason why these siRNAs tend to show perfect base pairing against their targets is because they came from those targets. The same virus that gave rise to them, it's a perfect match to their sequence, right? So that's why there is perfect matching, and that's why also, the adaptive value of this as an evolutionary mechanism is that now there is no inhibition of that virus. Oh, I will prevent your translation. No, I'm going to destroy your mRNAs. Yeah? Cool. So I just added here to the statement, miRNAs and siRNAs differ in the way that they base pair with the targets and also how they are made. Questions or comments, anyone? Yes?
So you're saying that siRNA of the virus is attacking its
So the question here is the siRNA of the virus is attacking itself? I would say that the siRNA is not of the virus. The siRNA is of the cell. It is the cell that chopped up an RNA molecule, or viral RNA molecule, made a whole bunch of siRNAs from it, loaded its risk complexes. So now that siRNA is not the viruses anymore, it's the cells. It's like the cell says, I'm going to take you, chop you up, and I'm going to make a whole bunch of siRNAs that I will use to defend myself. Yeah?
So, this mechanism, is it like similar to CRISPR in bacteria?
Very similar. Very, very similar. So, exceedingly similar. Yes. Yeah. I don't think we can afford the time to discuss CRISPR, but essentially, for those of you who know a little bit about CRISPR, CRISPR is a big machine called Cas9 that can cut, but the difference is that CRISPR will cut DNA. So this is almost like a CRISPR for RNA viruses. Yeah? And there is no, unlike CRISPR, there is no memory. So if, let's say that the cell fights off the infection, gets infected 3 months later, it will have to restart the process. As opposed to CRISPR, the bacteria keeps a memory of it. Yes?
So is siRNA only specifically to degrade mRNA from viruses?
I'm glad you asked. That's going to take us another detour. No. But this is fascinating. No. Okay. Let me add a slide for that. How do I add a slide? No. Why is it that I add a slide? I thought it was easier. Okay. I got myself lost in the app. All right, so let's do this. Found a little space here. Do you guys know how to use this app well enough?
What happened?
Notability.
Notability?
Yeah, how do I add a slide? I thought it was easier.
Top
Oh,
right.
here, add page. I got it. Yeah, thank you, though. There we go. Okay. Also, asked, is this mechanism always for attacking viral mRNAs? The answer is not always. We think that it evolved as an antiviral mechanism, and as it's so common in biology, once the mechanism was created, life found other uses for it. Right? So I asked you, what does a long double-stranded RNA mean for a cell? And I believe it was Jacob said virus, and that's true. But there is another source of long double-stranded RNAs in eukaryotic cells. Not very common though, not very common. But once you see this, you can't unsee it. It's beautiful. You guys ready? There are some genes. If I ask you to draw a gene, that's the line for DNA. I ask you to draw the TSS, the transcription start site, and convention is that we draw something like that, correct? So that means that there's going to be an RNA molecule that is produced from that TSS in a 5' to 3' direction. Turns out that certain species, for certain genes in them, evolved a secondary TSS that goes in the opposite direction. Right? Now, they don't turn on these TSSs. They don't turn on these particular core promoters, usually. But at some point, a cell may decide, you know what? I'm going to transcribe this gene, but backwards. And when they do that, what happens is now there's going to be a new RNA molecule that is in anti-parallel orientation and, of course, perfect base complementarity. So when a cell decides, and I emphasize decides, to do this, to express from both promoters for a given gene, it will automatically be producing long double-stranded RNA molecules. Once again, why would a cell ever want to do this? Now this thing, that beautiful mRNA that was produced originally can no longer be processed and translated and made into a beautiful protein. But what happens is this gets detected by dicer, that thing that chopped this up, right? So now this thing gets chopped up into pieces, right? So we start with this, and then chops it up into pieces. And each of those pieces can be used by the RISC to go patrolling as siRNAs to look for what? Well, it will find those guys really quickly and destroy them. Why, right? Why? Well, because remember, guys, cells decide to turn genes on, but at some point they may want to actively turn them off. At some point, all cells may say, I don't need this anymore. Actually, I really don't need this anymore. And they will turn that expression off. And intuitively, one might say, well, don't transcribe the gene anymore. Just turn off that promoter, that TSS, and you're done. Yeah, but these things are purposely made stable. They have a cap, they have a poly-A, they have RBPs protecting them. So they could hang around for a while. And for as long as they're around, they could be translated into a protein. And what if that protein became, for that cell at that time, toxic? So they may want to stop production of that protein pronto. I say pronto with my English accent. I just say pronto. Yeah. Right? So, is it always for double-stranded RNAs and RNA viral mRNAs? No. There's a few exceptions, and the thinking is that this was co-opted later, and since siRNA existed as a mechanism to defend cells from viral attack, some genes evolved these backwards promoters, and it became like a super quick off switch for them. Yeah? All right. This time, I think we should make it on time. So, cool. That was control of RNA stability through MI and siRNAs. Let's say that now we have a mature mRNA in the cytoplasm that has not been degraded by MI or siRNAs, or it's not being inhibited by them. So the next natural thing would be for them to be translated. And cells, once again, can regulate that process. They can decide when a certain mRNA will be translated. Super quickly, sometimes cells produce an mRNA and go through all of this trouble, but they don't want to translate those mRNAs yet for a number of reasons. One that is particularly important for the conversations that we will soon have. Think of a neuron, right? A neuron will express its genes in the nucleus. And all those mRNAs will come out of that nucleus that happens to be in the soma of a neuron. If you guys remember your super basic biology, a neuron will have a soma with a bunch of dendrites and then one axon that communicates to other cells, other neurons or other cell types far away, right? If an mRNA that a neuron produces is important for the terminal synaptic button, for instance, what's the point of translating it here? It's really hard to move a protein from the soma to the tip of the axon. It's much easier to move around an mRNA, right? So cells are constantly making those sorts of decisions. I'll make the mRNA, I'll have it ready, but I'm not going to translate it yet. So translation initiation is a highly regulated process. To understand some of the mechanisms, let's quickly review how initiation normally proceeds. So there is a bunch of proteins, as shown here, that will recognize the 5' cap. These proteins belong to the family of EIF. That stands for eukaryotic initiation factors. Right? So eukaryotic initiation factors. And the family is EIF4. So member E will recognize the cap, G will recognize E bound to the cap, and so forth. Simultaneously, there is other members of the EIF4 family, namely EIF4A, that can recognize, I oversimplified it there. I drew or make it look like it recognizes the poly-A tail. What it does is it recognizes and binds to the poly-A binding protein, PABP. So PABP is sitting somewhere here in the poly-A tail, and EIF4A can bind to PABP. When that happens and all these guys get together, that's a signal to the cytoplasm saying, yeah, this mRNA is whole. I can see a 5' cap and I can see a poly-A tail. So it's relatively safe to go ahead and translate this thing because we know that it hasn't been truncated anywhere. If an mRNA has been truncated somewhere such that the 5' cap is missing or the poly-A tail is missing, how does the cell know if the message for the protein is whole or not? So that's a humongous waste of resources if it turns out that it wasn't a complete protein. So cell won't take any chances. It will initiate translation only of the mRNAs that are complete. And when all of these things are in place, the EIF3 and EIF2 factors, additional factors, so the eukaryotic initiation of translation will join the party, along with the small ribosomal subunit that comes preloaded with those very special tRNAs that can recognize start codons. If you guys forgot your translation days, AUG stands for methionine, so the very first amino acid in all proteins will always be methionine. Whether or not that methionine stays in that final protein, it's a different story. But they all start with methionine. So when everybody is in place, the initiation complex will start scanning that mRNA in search of that AUG. And that's where the Kozak sequence that Josh was bringing up before comes into play. So they won't stop at just any AUG. They will stop at an AUG that is in the context of one of these consensus sequences that we call a Kozak sequence. It's almost like the mRNA is telling the small ribosomal, all right, we're the Kozak sequence. Now, listen up, because when you continue scanning, the first AUG that you find, that's the one. Okay? So this thing goes through the mRNA, finds the Kozak sequence, acquires some sort of conformational change, and then it's ready to see that AUG. And when it finds it, that triggers a conformational change in the small ribosomal subunit such that now the large ribosomal subunit can latch onto it, form a whole ribosome, and now translation can proceed. Okay? So that's how things happen normally when an mRNA is immediately translated. But cells can delay this process, as I was saying before. Sometimes they don't want to translate an mRNA right away, right? So one mechanism they do this is by sending a protein that is called EIF4BP. What do you think BP stands for? Binding protein. Binding protein. Yeah, BP stands for base pairs, but upstream. Binding protein. Anytime that you see a BP, chances are that it's going to be binding protein, like PABP or RBP. And we mentioned two today already. So EIF4BP is a binding protein that binds the EIF4 complex, the one that assembles at the beginning of the mRNA, making sure that there is a 5' cap and a poly A tail. And this EIF4BP will serve as a physical block to the initiation complex. It won't let it start scanning. Okay? The only way that this initiation complex can start scanning is if the cell says, all right, now we can translate this thing. So what will the cell do? Phosphorylate, each of these red dots indicates a phosphate group that is added enzymatically to this binding protein. All of these new charges will make the EIF4BP undergo a massive conformational change such that it can no longer hold on to the EIF4 and it falls off that mRNA, relieving the block on the initiation complex that can now start scanning that mRNA. All right? I can't remember with whom we started this discussion last week, but at some point we mentioned that depending on, say, the nutritional state of a cell, cells may want to express certain genes or not. And we said that way that a way that the cell has to sense nutritional states is by phosphorylating or not, or expressing or not certain transcriptional activators, right? And I stand by that. Cells do that. They will, if they're starving, they will not express certain activators, or they will phosphorylate certain activators in ways that won't allow them to do their job. So certain genes won't be even transcribed. So I stand by that. The nutritional state feeds primarily through this mechanism. And it's, again, a very elegant, very interesting mechanism for expression regulation. And it goes something like this. Cells evolved to understand, quote unquote, that starvation may be temporary. It may be just a transient state. We're starving now, but who knows? Any time there could be a flood of nutrients and energy, right? So if we need to start transcribing genes when food comes in, it will take a while until we have our protein. So what many cells slash organs slash organisms slash species evolved are mechanisms by which a whole family of mRNAs is transcribed, processed, and is ready to go, but they're blocked by this eIF4BP. Because eIF4BP cannot bind to just any mRNA. They will bind to mRNAs that have sequences that they themselves recognize. So eIF4BP is a binding protein for the eIF4 complex, but it is also in a way an RNA binding protein that can read certain sequences in certain mRNAs. So why would cells do that? Well, it's relatively straightforward. There may be mRNAs that the cell wants to have ready for when food comes. And when food comes, they don't want to wait until the thing is transcribed, spliced, exported out of the cytoplasm, polyadenylated. Those are many steps. So they will have the mRNAs ready, and then when food comes, there is a bunch of kinases that will heavily phosphorylate eIF4BP. Now eIF4BP falls off of those mRNAs, and now these mRNAs can be translated. There's many examples. Let's go through one. This mRNA, for instance, might code for an enzyme that is important for metabolic reactions that will store energy. So these may be enzymes that participate in making glycogen out of sugar, out of simple sugars. Why would you express that enzyme when there are no sugars around? There's no point. But cells evolved a mechanism by which the second sugar shows up, we want to have that enzyme ready. Right? So they block the translation of those mRNAs, but when sugar shows up, this thing gets heavily phosphorylated, the enzyme is quickly expressed because everything is ready to go, and now the enzyme can take all of that sugar and turn it into glycogen that can be stored in the liver. If this is a plant cell, it would be starch or something like that. Yeah? Cool? All right, and last but not least, another mechanism by which cells can sometimes control translation is by hiding the Kozak sequence and the AUG. And I want to speed this up a little because I want to be respectful of the time, but I have a question there. How can a cell hide the AUG? Anyone quickly? Jessica? Exactly, through secondary structures, I think that I have, so either through secondary structures that include the Kozak sequence and the AUG, and or, because it could be either of those mechanisms, specific RNA binding proteins that are blocking them, a certain mRNA may not be showing its Kozak sequence and AUG, and it will be invisible to the ribosome. Okay? Cool. So last but not least, and we don't have much time to go through this slide, but I think you've touched on this. We touched on this in previous weeks, but the idea that once a protein is translated in the ribosome, for most proteins, that's not the end of the road. For most proteins to acquire full biological function within the cells that they're expressed or the organism they're expressed in, more often than not, they need to undergo a series of post-translational modifications. And by the way, if you guys engage with those reviews that I share with you for Alzheimer's, you'll see some references to PTMs. And as long as I could tell, they were not explained anywhere. PTM stands for that, post-translational modifications. And what that is a series of chemical reactions that are done, chemical modifications that are done to key amino acids in the polypeptide sequence or the protein sequence, such that now these proteins will have to refold in space to accommodate to that new chemical reality. And by refolding, they will be acquiring new shapes, and therefore they may be either acquiring or losing function, right? Because the function of proteins is very tightly related to their shape in space. There may be chemical modifications like palmitoylations or myristoylations that attach lipids to proteins in such way that now these proteins can anchor themselves in cell membranes. in the context of Alzheimer's disease, you'll read in your reviews that a very important protein in Alzheimer's called ApoE is lipidated this way. So now the ApoE protein has a bunch of lipid anchors hanging out from it. There is strategic proteolytic cleavages that will chop out in a somewhat irreversible manner, as we discussed before. It will take out certain regions of a protein, certain segments of a protein, making it now active. So the example here is insulin, but another classic textbook example is clotting factors. They're all throughout your bloodstream right now, but they're inactive. Otherwise, we wouldn't be having this conversation, right? We'd freeze in place like all clotted. But when there is injury, there is a bunch of enzymes that will go and strategically cleave these pro-clotting factors in specific places, activate them, and now the clotting cascade begins. And finally, we talked about how a cell may at some point say to itself, I don't want to express this protein anymore. So it may stop transcribing it, it may stop translating it, but at some point, if it really wants to get rid of that protein, it also needs to destroy whichever proteins it already has. And for that, there is a very regulated mechanism that will mark proteins that need to be destroyed through a chemical reaction that will add ubiquitin molecules to those proteins. And once a protein is ubiquitinated, meaning that it has all of these ubiquitin marks associated with it, there is this gigantic, basically recycling plant that cells have that will take these proteins and chop them up into individual amino acids. Okay, so that's the final turning off of a protein, when not only am I not expressing it anymore, I take the ones that I have and I actively destroy them.